So previously, I talked about some basic principles of genetic toxicology. And okay, as soon as I get my, all right. So I just wanted to get going because get myself talking here. I wanted to point out, like between CIT, where I was doing basic research, I went to work, after they let us go and I went to work for Merck Pharmaceutical. So I went from a basic research environment to a highly regulated safety assessment facility at Merck. And Merck safety assessment had 480 something people. They were Part 11 compliant, crazy. And I ran the Ames tests for all the products coming in for Merck. And at the time, we were testing about 80 Ames tests a year, probably 45, 50 new test articles. And basically, it was kind of a little bit of a rude awakening because I went from being able to publish when I was at CIT over those 13 years, published something like 60 papers. And it went from, can you publish, to, do you know how to use Excel? And so just to recap last time, I want to make sure that there's a known difference between genotoxicity and mutagenicity. Genotoxicity is something that happens early on, 4 to 24 hours after exposure. You're just looking for DNA damage or gene expression profiling or DNA adducts. Also, micronucleus assay, chromosome aberration assays. These are measures of genotoxicity. They are not measures of mutagenicity. Mutagenicity takes cell proliferation, takes cell growth, and it takes a while for that old non-mutated allele to be converted to a mutated allele. And that typically takes somewhere between 5 and 7 days after exposure. So all these things that we're measuring early on, COMET, any kind of toxicogenomics, all that stuff disappeared. And what you're looking for is a cell that got damaged, but is able to faithfully replicate its chromosomes and produce daughter cells. So when we do Ames tests or when we do these assays, the cytotoxicity is based on mitotic index, how many cells got to mitosis and made metaphase chromosomes, so we can score them from aberrations. Are the cells growing relative to untreated controls or clonal growth? Those are the only acceptable measures of cytotoxicity. This is genetics, and you got to show that that material is being passed on from one generation to the next. ATP, MTT, all these other assays that are out there, aldehyde dehydrogenase, none of that tells you whether that cell is going to survive that insult, damage, replicate its DNA, at least get to metaphase, one cell division, or better, can they continue to grow and form mutations that can be scored. So the genetic toxicology test battery, this was first came out in 1997. It was revised in 2012. I forgot to spell out ICH. That stands for International Council of Harmonization for Pharmaceuticals. So more long-winded to that. But it's an organization that decided that instead of having each country or nation like the United States or Europe or Japan have different gene tox testing requirements, this is an attempt to harmonize all the testing requirements. And they are now in 2012, this is what's ruling the day now. They were given two options. Option one includes a bacterial test for a mutation in bacteria. That's also referred to as an Ames test, which is, we also do, the test number 2 is a mammalian cell test. It's measuring genotoxicity, either chromosomal damage or in vitro micronucleus, or the in vitro mouse lymphoma assay, which is one of the TKG mutation assays, the G mutation assay. It's the only mammalian cell G mutation assay, and it's the only one that's used in this DABT battery. And finally, you go in vivo test for genotoxicity, usually a chromosome aberration test. They also provided in this guideline option 2, which eliminates the mammalian cell in vitro test, number 2 there. You go from an Ames test directly into animals. And then the animals is where you do the micronucleus and COMET assay. And I'll talk about those in a minute. And it had to do with the fact that these mammalian cell tests in vitro, this was 2012, these were a bit of a mess. They had a high false positive rate, and there's a lot of concerns about them. And this is this document came out before the revisions to the OECD test guidelines. So about 2012, myself, I was asked to be a part of the OECD expert working group that revises genetic toxicology guidelines. And it was a rather eye-opening experience to learn how much effort it takes to get one of these OECD guidelines to either change or to add a new one. And this is a huge problem. It takes years to do. All right, so I did want to mention, we do the Genotox testing here. And so I wanted to put those tests here and tell you who kind of runs these in the group here. So the Ames test is under the domain of Carol Schwartz, Jamie Sly, so Sarah, Caitlin, and Alex all help out. The micronucleus tests, in vitro micronucleus, is run typically by flow cytometry. So Nick, Christy, Jasmine, and Lincoln are able to run that test. And we also do an in vivo test for genotoxicity, and that's Mike Stryker is typically the state director for all these tests. He's supported by Comet work as also the micronucleus, and that's Nick, Jamie, and Lincoln again. And when we do GLP studies here, we test the dose solutions to make sure they're accurate. And so when we do GLP studies, we got to test the dosing solutions for stability, uniformity, and accuracy. And that falls under Keithberg and Ivy. So if you guys have questions about something and you want to see these being run, please contact these folks. I'm sure they'd be happy to show you what's going on. And finally, one thing before I launch into these, one of the issues that comes up is none of these cell lines have the capability of metabolizing compounds. As when you take a drug or something, it's metabolized to be basically converted from a lipid-soluble drug to a water-soluble product that can be excreted. And this happens by drug metabolizing enzymes. P450s play a key role, cytochrome P450s, but phase 2 enzymes also, in my view, have a more critical role. But one thing that we use to accomplish this is so-called rat liver S9. And so I put this slide in here because people are using this as a reagent, and I want to make sure they know what it is. And so with rat liver S9s, you take a rat, you dose them with with phenobarbital and beta-naphtoflavone, which are highly induce cytochrome P450s to an incredibly high level. So they are administered this for 3 to 5 days. Take the liver, you homogenize it up, and you you spin it in a centrifuge, and the supernatant from a 9,000 G spin is S9. So it's called S9 because it's a supernatant from a 9,000 G spin. When we use it in short-term tests, it's only supplemented with NADPH cofactors, which drive cytochrome P450 mediated metabolism. And so you don't have phase 2 metabolism in these systems, and that's a huge problem, at least in my mind. I've got a grant on that basis. So what happens when you give animals this, their liver weight increases, P450s get upregulated tremendously, and that slide on the right, picture on the right there was done by Miriam Rivas, and you can see that cytochrome P451A1 in this system is induced about 5,000-fold, and CYP1B1 1,000-fold. You have massive induction of cytochrome P450s. This is also being used in short-term tests and high-throughput assays as well, and I think it's going to be a problem, particularly when we start using those data for quantitative benchmark dose, because it's a highly misleading way of drug metabolism. Humans don't metabolize like this. All right. So when you do a Genotox test battery, we're measuring 2 types of events: a gene mutation, single changes within a gene, and also chromosome aberrations. Well, why is that? Well, because some compounds, it can induce both, like those shown here, but some are very, very specific and only induce, for instance, chromosome aberrations, something like benzene. There's very little data showing that benzene causes gene mutation, but a lot of data showing that can cause chromosome breakage, and it has to do with mode of action. That's why when we do a Genotox test battery, we run an assay for mutation, which are quote small events, and an assay for large events, which are chromosome aberrations, which involve millions of base pairs. And this, and there's also one other document that's used to go look at is this ICH M7, and I think this is kind of white paper, so to speak, on how gene tox testing will be done. This is, in the case of a drug, you have to have 98.5% impurity, pure drug to be tested. If there's an impurity that's greater than 1.5%, it needs to be qualified. And what does that mean? Well, it means be qualified to define what's called a TTC, threshold of toxicological concern. This is used in gene tox, and basically the important part is just down here, where you do classification of a material like a impurity. You do QSAR analysis, an Ames test, and some potentially in vivo follow-ups. And I think this model of combining computational methods with in vitro testing is basically how the future is going. And to talk about computational methods, this is there's all this interest in QSAR. Well, this was one of the first papers, and maybe the first paper, I'm not sure, you get trouble when you say this thing. It's a fellow by the name of John Ashby in 1989. And he was a consultant for the NTP. He worked for Imperial Chemistry, ICI in the UK. He came up with this structure, and he, along with some other folks looking at the NTP databases, decided that they're so-called structural alerts. And these structural alerts can be used to predict potential genotoxicity. And this is the computational method. He drew the structure. He's a chemist. I'm not sure if I think it's supposedly accurate. All the double bonds are correctly, but it's got 5 bonds, no carbons with 5 bonds. But anyway, this just to give the idea that nowadays with these software packages on the right here, Derek, Multicase, LeadScope, they all can do these kind of what they call assess for structural alerts. And these software packages use 2 kind of methods. One is called the expert ruling, and the second is called statistical ruling. And you need both to make a call with respect to, is this compound have a structural alert that can use to predict a potential outcome in the Ames test. And they can, quote, predict other outcomes, but it gets pretty bad as you go up in things like, you can predict all kinds of things, but whether it's real or not. But typically for the Ames test, it works pretty well. And one other point about gene tox testing, this is one of the things I learned at Merck. For a phase 1 clinical trial, you need to do an Ames test and an in vitro micronucleus or some kind of in vitro assay for phase 1 clinical trials. For phase 2 clinical trials is when you start doing in vivo testing. So first round of drug development, phase 1 clinical trial, no in vivo gene tox assessment. However, there is both a rodent and non-rodent species assessment of toxicity. All right, so the Ames test. What is the Ames test? And the Ames test was something I did when I was undergraduate. I was a senior microbiology student at the University of Kentucky, and I had to do a senior project. And I met some guy. He told me, there's a test. You can tell whether something causes cancer or not in 2 days. And I said, well, what is that? And he told me it was called the Ames test. And it's kind of remarkable that a test that was developed in the late 70s, 78, 80, early 80s was kind of when it took off big time, early 80s, that this test is still used today. And it's actually some remarkably engineered bacterial cells that enable this to happen. So what happens in the Ames test? We have the Ames group, Bruce Ames and colleagues, developed some histidine requiring Salmonella strains. And basically, the story goes that he was eating a bag of potato chips one time and was looking at all those ingredients and wondered, gee, I wonder if these are harmful. And basically, he did the Ames test and he developed the Ames test. And this is, this is a reverse mutation assay, because you go from cells that require histidine to cells that do not require histidine. So you're mutating them to that form. So it's a very simple test. This is one reason it's still being used, is you take the cells, you mix them with rat liver S9 or not. We do both with and without testing. And then you add your test compound, you put them in a soft auger, you mix them up, and you pour them on a plate that has very little histidine and very little biotin. And biotin is kind of irrelevant to talk about later, but the histidine is important because, as you guys remember from last week, to cause mutations, you need cell proliferation. So when we do this, we add a little bit of histidine because these cells require histidine. So that enables them to go through those first few rounds of replication to fix the mutations. And then now, after 2 or 3 days in an incubator, they form colonies on a plate where previously they did not require, they required histidine, now they don't. So that's basically how the Ames test works. There are commonly used strains that are used in the Ames test. The first set are the TA numbers. Those are all Salmonella. And so Bruce Ames created all the Salmonella strains. The E. coli strains on the bottom were created by another guy whose name was Green, Green, and he was from the UK. So these are the tester strains that are used. So there's the mutation, the type of mutation, the main DNA target, and then also the plasmid over here, whether they have a plasmid or not. So if you look across here, we typically use TA100, TA98, TA97A, and I think 1535. And so you look across these, you see that the target for most of these is GC base pairs. And so what would happen is that when this became apparent that these are all GC base pairs, someone thought, gee, we need a mutation assay that detects AT base pair mutations. And so this was happened with TA102. And then these E. coli strains here were developed as well that detect mutations at AT base pairs. So when you run an Ames test, you run strains that have mutation that can detect mutations at GC base pairs, and one strain that can detect mutations at AT base pairs. And finally, at the very back here, this plasmid PKM101, it's named after a woman by the name of Kristen Melmans, and it has what's referred to as SOS repair. And SOS repair, I think, was discovered by a woman by the name of Evelyn Whitkin. And I think she won a number of prizes for this, but it was the first demonstration of an inducible response to DNA damage back in the 60s. Evelyn Whitkin showed this with UV light, and she dubbed it SOS repair. And exactly what it means that when a bacteria is confronted with DNA damage that it cannot fix, cannot replace, it induces this DNA repair response that just puts in any base pairs in there and fixes it, just willy-nilly, so to speak. And that is referred to as an error-prone DNA pathway. So when we do, so this is a plasmid that is in these strains, and we do testing with and without the plasmid. So basically, and you can see the effect of the plasmid, for instance, in ones without plasmid versus one with the plasmid, the background frequency is about 2-fold higher in the ones with the plasmid, just by the fact that it's inducing error-prone repair and repairing spontaneous DNA damage in an error-prone manner. So that's how these strains are set up. And this is one reason when you see any, so this is from OECD 471. So when we do this test, we test it to 5 mg per mil, 5 mg per plate, I mean. These are cell plates, petri dishes. We do 5 doses with and without S9. We do positive controls, and there are some positive controls that are kind of diagnostic for each strain, so they respond to some compounds differentially between the strains. And for a positive, we use what's referred to as the 2-fold rule, for the most part, sometimes a 3-fold rule. So you got to have a dose response, a fold increase, and you have to exceed the historical control range. And the point about different strains, this is why a positive in any strain is considered positive. So this is when complaints say, well, you got to know, they each detect different types of DNA damage. So in each strain, it's a unique system. And so any strain that's positive is considered a positive. So as I said earlier, if there are questions, Bill, you can unmute and ask me a question, by the way. Sorry to brought that up. So as I mentioned earlier, the mammalian cell tests were fraught with false positives. And OECD groups were formed in 2014 to come up with new methods or knew how to look at these. And so there are a lot of things that came up. One of the things that was interesting was the rodent cells that are used for genotox testing are P53 deficient. So you have a situation where the rodent cells do not have a good DNA damage response. and that can lead to false positive, because if you're not, responding to the DNA damage like a cell should, you're not going to fix the damage. That can lead to mutations that can lead to false positives. On the other hand, these human TK6 cells are p53 proficient, and they're human cells. So the human TK6 cells that are p53 proficient are becoming more widely used because they're human and p53 proficient. And just serendipitously, this cell line was developed at MIT. And when I was a postdoc, I worked for a guy from MIT, and he's on the patent for the cell line, or was on the patent. It expired many years ago. So I've been working with TK6 cells since 1986 or so. And so I'm very familiar with them and how they work. And that's one reason I was asked to be on the OECD expert working group, because TK6 cells were being put into some of the OECD test guidelines. Besides that, we have top dose levels that were pretty drastic. And 10 millimolar is what the top dose is right now. Still for OECD, for pharmaceuticals, for the mammalian cell test, 1 millimolar is the top dose, or a dose that is cytotoxic. So if it's non-cytotoxic, you got to go to 10 millimolar. If it's for chemical, if it's non-cytotoxic in a pharmaceutical, you got to go to 1 millimolar. However, more recently, with complex mixtures like tobacco products and things of this nature, they forget about concentrations, and the guidelines, or at least the European Food Safety Authority, is recommending that you test levels that cause up to 50% or so cytotoxicity. So we have these issues with false positives, and then finally the bottom one, which is what I've been able to capitalize on, is that a lot of these cells like drug metabolizing enzymes. What's used is S9, which is an imbalanced system because it only has phase 1 metabolism, not phase 2 metabolism, and that I can talk about that forever if you. All right. So that's the Ames test. The next test is chromosome aberration test. How do you do this? Well, we can usually start with either human lymphocytes directly out of people, or CHO cells. The human lymphocyte thing is less used now, and back in the quote good old days, you can go down the hall and grab someone and stick them for a blood sample and use it for the experiments. Can't do that anymore. So we have to, if we do this, we also learn you need fresh blood samples for this. So we try to, this is a concern because you have to get blood samples from people that aren't tested for any kind of viruses or anything. On the bottom side are Chinese hamster ovary cell lines, CHO cell lines, and those are very frequently used for chromosome aberration tests. And the reason is they grow good and they have big chromosomes. Big chromosomes, they have 22 chromosomes. We have 46. They have 22 and they're very large, or what we call metacentric chromosomes, which means the centromere is in the middle. So you got a nice big chromosome, you can easily score chromosome aberrations. So what happens is we treat the cells with or without rat liver S9, and then we stop cells from going into metaphase, and we catch them at metaphase, basically. And we use colchicine to stop them there, and then we put them on a slide, and then some person has got to sit down there and count how many chromosomes each cell has. We got to count 300 cells per concentration. And it becomes, you got to know how to look at chromosomes and score them. One of the problems here is it's really hard to tell if you lose a chromosome or not, because you have a certain, what we call modal chromosome number. For Chinese hamster cells, it's 22. So, can you really detect 23, so to speak, in a population? So that's how we do. This is very old school. It was done in Hiroshima for the, after the nuclear bombs. This is 50, 60-year-old test. However, we still get clients that want to do this. And it's kind of mind-boggling that in 2020, they still insist on using this. And there's a lot of reasons why, but this is in contrast to the micronucleus test. Some research scientists made the observation that when cells have chromosome damage, right when they go into metaphase and split into two cells, they have the main nucleus, which contains the majority of the chromosomes, but if there's a fragmented piece or a chromosome missing, that gets encapsulated in what's referred to as a micronucleus. So this was a big discovery because you were able to basically take cells and take mitotic cells and then score these down here. So it's basically, these are biologically equivalent to chromosome aberrations. You can do this manually, or you can actually do it by flow cytometry. And again, we still have clients that still want manual scoring, which is mind-boggling. But this, and there is a cancer risk now for classigen. There's theoretically linear extrapolation, I think, which is being challenged by folks like myself. Don't believe that kind of thing, or a threshold, which I think is I don't know if I want to use the threshold, but you can invoke a safety level with an antigen where you can't theoretically with a classigen or a mutagen. You have zero, basically linear extrapolation of risk. Chromosome aberration versus micronucleus scoring, you can tell the difference. You look at the left versus the right, which one do you want to score? And basically, this is all manual scoring. Some clients still expect us to do manual scoring because that's how they've always done it, kind of thing. But this is labor intensive. We do have a cytogeneticist, Patty Allen, that we bring in occasionally to count chromosomes if we absolutely have to at the request of a client. We try to persuade them to use a micronucleus test and to use basically a flow cytometer. But sometimes this is an uphill battle, and one unexpected thing is some of our competitors would tell people, even though it's written in the guideline, that the micronucleus assay by flow cytometry is not acceptable by the FDA. And when you have a competitor telling that to clients that you're trying to court, sometimes it makes no. So how do you follow up the positive? There's also this newer assay called the multi-flow assay. And this is a biomarker-based assay, which uses a number of biomarkers, including nuclear P53, to assess whether something causes classigen or an antigen. So this is how you assess whether these are aberrations there. However, the bottom one does not meet OECD test requirements, so you really can't use it. You can use it in drug discovery, but not in preclinical safety assessment. So the mammalian gene mutation assays are shown here, and these are a bit of a pain, and we are setting one up called the mouse lymphoma test. And basically, there are two guidelines. I was on this one, 490, because of my TK6L experience, and also in the general group. So where do these assays work? Well, you're measuring, these are 4 mutation assays. You're going from an HPRT plus mammalian cell to an HPRT minus mammalian cell. Or it's a thymidine kinase gene heterozygote, which is an autosomal locus, versus forming a TK minus, so you're losing that plus allele to mutating it to a minus allele. There's only one plus here because this is HPRT's X-linked, going to minus. These are 2 alleles, this autosomal locus, chromosome 17 in mouse, and I think chromosome 11 in rat and humans. The selective agents for these mutations are 6-guanine for HPRT and trifluorothymidine. So what these 6-guanine does is it gets incorporated to the DNA and kills all the wild type cells. And basically the cells that are mutant survive, and you can count them as colonies. Same over here, the trifluorothymidine gets incorporated into the DNA. It's a false DNA substrate. The cells that incorporate it that are wild type that can incorporate it die, and the ones that don't incorporate it because they've been mutated by your exposure, they are survive. So this takes 6 to 8 days for this to occur after initial treatment. Over here, it takes 2 to 4 days. So the difference in time between these 2 alleles and the endpoints are colonies. So you need to treat large numbers of cells for this, and this is why there's no such thing as a high throughput mutation assay. And there might be one coming along soon, but I'll talk about that later. So why is there no high throughput testing assay for mutations? Well, because you need to treat here. When we do these experiments in the past, we treat 4 times 10 to the 5th cells per mil for 50 and 50 mils in a T75 flask. That's 20 million cells. Well, why do we do that? Because the mutant frequency is 1 in a million, 1 in 100,000. So you need to have a population that size to make sure that in the flask that you're treating, there are a number of pre-existing mutants. And so in our case, we want to have somewhere between 20 mutants in our flask. Before we start, these are spontaneous background mutants. And then when we treat them, we have to allow them to grow. So basically, as I said last week, this takes a number of times. So cells grow, you treat a cell, and if this is not repaired and you get a survivor, it can form a DNA damage. Then the next cell division, this daughter cell has a heteroduplex, basically an A pairing to a C or a G pairing with an A, so to speak. However, the rest of the cells are, that other daughter cell is normal. It goes on and grows. However, this mutated cell keeps growing and forms eventually a both allele affected mutated cell. This takes at least 2 cell divisions and again, 3 to 7 days. So this is not high throughput. You can't make it high throughput because it's genetically impossible. And so this is why, for instance, there's a new technology called duplex sequencing. And I put an SBIR grant in it because this can be done now in theory by DNA sequencing directly rather than wait this amount of time. And it's interesting because the vast majority of the data we're collecting in mammalian cells are assays that suggest that there's a mutation there. There could be, you've got chromosome damage, you got micronucleus, but that doesn't tell you if that cell survived that, formed a daughter cell that now are carrying mutations. So you got to have a cell that's been injured somewhat, but it's still viable, can keep chugging along and form daughter cells that are mutated. So again, OECD guideline 476 and 490 cover these assays. I put that in blue. This was a huge argument. We're talking about doing experiments where you're killing 80% of the cells, maybe 90%. And you're also testing at 10 millimolar. So there's a lot of issues here that could be ripe for a potential false positive. And finally, you grow and select, and then finally, at the end of the day, for certain locus, the thymidine kinase locus, you form 2 kinds of mutants. One are referred to as small colony mutants, and these are believed to be due to TK mutations in that genome, within that gene, so to speak. You also get these slow-growing mutants, which are mutated outside of that genome. So the idea is the large colony mutants are normal growing, they have a mutation in the TK gene. It really doesn't affect them that much. They keep growing fine. However, the small colony mutants have mutations that incorporate not only the allele you're looking at, but alleles downstream and upstream that can compromise the cell growth. And it becomes important because the small colony mutants are a lot of these are where you have a lot of false positives. So you got to be able to score these and look at whether, I've got a lot of small colony mutants. Is this really a positive response, or is it just due to cytotoxicity? You're going up to 80% killing, 50% killing. Is that small colony due to just damage in the genome? Cells can't grow very well, that kind of thing. The in vivo micronucleus assay. Now we're going in vivo, and basically, there are 2 ways to do this: single cell treatment, and then you collect the bone marrow 24 to 48 hours later, because again, you got to wait after the damage occurs. Same thing in vivo in the bone marrow. You got to wait for cells to go into mitosis and go into cell division. And for the micronucleus assay, because you're looking at daughter cells, you're not looking at the cells. You got to give that some time. Or you can do daily treatments and collect bone marrow 18 to 24 hours. The other way to do this, which is the easier method, is to collect peripheral blood. And in that case, you need to wait 36 to 72 hours or 36 to 48 hours after the last dose to collect blood to measure the micronucleus. And basically, you can do this by microscopy, where you basically take a slide and you have someone sit down and count micronuclei using a microscope. Or again, you can use flow cytometry. And you can, it takes, half an hour here, it takes about 2 minutes over here on the flow cytometer. So why do we look a day early? Well, a day later or 2 days later after exposure for the micronucleus test, if you're measuring blood. Well, it has to do with the fact that the DNA damage that you're measuring occurs in the bone marrow. So in the bone marrow, there's a hematopoietic stem cell, and that cell sustains certain DNA damage. It forms erythroblasts, which are early red blood cells that contain nuclei. Red blood cells, at least human, don't contain nuclei. So what happens is, as that cell is maturing from a stem cell, which has a nucleus, an erythroblast that has a nucleus, the main nucleus gets extruded. So all the bottom part, this is what happens normally. You get a nucleus extruded from a red blood cell. This happens in the bone marrow, and then as it goes and exits into the peripheral blood, you now have a cell, a red blood cell, a normal red blood cell on the bottom here, that has, in one case, it forms a reticulocyte, and that's an early version of the red blood cell. It's also called polychromatic erythrocytes. And the reason it's called polychromatic is they still have mRNA in them. So when you add DNA stain to see the DNA, the nucleus, the micronucleus, that stains as well. So you get this pickled stickle pattern, and it's called polychromatic. And that's compared to a nomochromatic erythrocyte, because there's nothing in there that will stain with a nuclear stain. If you go on the top panel, though, we have DNA damage. Again, the cells divide, they form micronuclei, as I showed earlier, just like in vitro. But what happens is the big nucleus gets extruded and the micronucleus stays behind. So basically, you're able to, and this is happening in the bone marrow as it exits into the peripheral blood a day later, you can see micronucleated reticulocytes. Again, these are immature red blood cells, still have RNA in them. And then you can also see this in the fully mature red blood cell, which takes basically another day to go from here to here. So basically, how we do this is, you can either score these by microscopy, but a flow cytometer is pretty slick because we can do antibody staining to CD71. So we quantify CD71 plus cells, which is a cell surface marker on reticulocytes. So we're gating for C71 cells and cells that contain mDNA. So you do a double analysis on the flow, CD71 and DNA stain, and that tells you there's a kit to do this now, and it's micronucleus. So micronuclei are produced by chromosomal aberrations in aneuploidy. Elevated micronuclei are shown to be related to cancer risk. And then and again, it's not a huge, it's a rather shallow slope, but there still is a risk for that. And the interesting part of this is you can use this across a number of species, even in humans. So as guidelines say, well, again, you harvest cells after treatment. If you're using peripheral blood, you want to look a little later. We typically use a maximum tolerated dose of 2,000 mg per kg in animals. If it's not cytotoxic, we always do a rangefinder for these. And then 4,000 immature erythrocytes are scored per animal. And by flow cytometry, we've typically scored 20,000 cells in the past. When you do a rangefinder, you do males and females, and you use the sex that responds the best or is most toxic to. If there's no sex differences, you can use 5 males, and there's some push now to use 3 males and 3 females in each dose group. And you can integrate this with repeat dose testing. So with the NTP, for instance, we're doing the micronucleus assay on 90-day studies, 28-day studies. So it's very handy. You can just basically, all you really need is a blood sample from an animal. And we typically use only 200 microliters. So from a rat, it's fairly doable without compromising a 90-day study, for instance. The common assay. Well, the common assay is also referred to as a single-cell gel electrophoresis assay. It's basically an assay that was many years ago called the alkylation assay, and it's basically a miniaturized version of this. And it turns out when you have put DNA at a pH greater than 13, it becomes very fragile. And what happens is if you have a DNA adduct, a DNA lesion, or you have what's referred to as apurinic sites where you've lost a purine, this can be converted into double-strand breaks and single-strand breaks, which then you can detect by electrophoresis. So Common assay detects a broad range of DNA damage and lesions. Some people think it's just a breakage assay, but it's not really. Again, well, if you have a DNA adduct at alkaline conditions, becomes unstable, forms a break, and then that's how those are scored. The advantage of the common assay, unlike the assays I talked about, chromosomal aberrations, you got to have cells dividing. Well, the common assay, you don't use a cell that's dividing. So this is why we use the bone marrow and the micronucleus test, because we need a cell population that's constantly dividing. In the common assay, it's completely opposite. You can do any tissue, brain, liver, lung, any tissue, because you're not worried about cell division, because that's not part of the assay. It's you get single cell data, and it's sensitive. I put it in a question mark, and it's economical. Its disadvantages: it's low, tedious scoring. You got to have harmonization. You have necrosis, genotoxicity that can be a confounder. So when we run the test, we also put in, make sure they know, we put in money for histopathology. So in case there might be a confounder by cellular cytotoxicity. And the sensitivity thing is one of my pet peeves. Everyone talks about how sensitive the common assay, but if you think about it, the way I think about it, if you look at a liver, you have 100 million cells in a liver, and when you do the common assay, you're sampling 150 cells. So out of that 100 million pool of cells, you're pulling out 150 and looking at those. Well, off the bat, that tells me that among those 100 million hepatocytes, you got to have pretty extensive damage if you're only going to go in there and pull out 150 cells and analyze those. So when you say sensitivity for the common assay, well, they typically we can measure in any tissue. Well, in sensitivity with respect to, is how damaged is that tissue? It's got to be pretty damaged to be able to measure a signal when you're only pulling out 150 cells, 200 cells to score. So how does the assay work? You take cells from either an exposed animal or cells from in vitro exposure. There is no in vitro common assay, so there's no guideline for that. It doesn't exist. But the one for the in vivo assay, take the animal and basically we mince cells to make single cell suspensions out of, for instance, a liver or any tissue. We also have used a sieve. And the idea is to be able to make a single cell suspension from that tissue, brain, liver, single cell suspension. You take that, you mix that with agarose, and you put it on a slide. So now you have cells in agarose on a slide. You take that slide and you remove you do cell lysis, get rid of all the cell debris, unwind the DNA, and what remains is little DNA balls from the nuclei embedded in the agarose that are remnants from a cell. So you have little DNA, nuclear DNA sitting in agarose, and then you do electrophoresis. And then, so as the DNA gets fragmented, you get more and more DNA damage, and we can do this by imaging, shown here using this imaging system. This is shown here again, the imaging system, scoring these by image analysis. And then it requires, so this is one at a time, click one at a time, 150 cells, one at a time clicking. And basically, it's okay to do this, however, it's very difficult to do, very labor intensive. And the problems are shown here. You score these at the bottom. So you look across here, you get percent tail DNA, which is the metric that's being used. It literally is, if you go back to this slide, how much percent of the DNA is in this tail? This is all done automatically by image software analysis. So you get to the point where, gee, we're scoring this one at a time, and then do you score this? Do you not score this? There's also what's referred to as hedgehogs. And these are cells that are very damaged that you don't score, but we have to tabulate them as per the guideline. And just for way of reference, we did get an SBIR to develop Comet Chip, and this is an automated version of this assay. So each one of these wells, like A1, has a few hundred wells in it, and basically you're able to do single cell arrays in this technology developed by MIT. And so instead of doing one at a time, as you would do in the regulatory assay, in this assay, you can score, you can image each well, and then you get this picture over here and you can score it automatically. However, you can't do this right now with the traditional COMET because it's not written into the regulatory assay. So with the COMET assay, you basically you're scoring the 150 cells per tissue. Percent tail DNA is what we use. And it's interesting, this little metric here, when we do the COMET assay, we use the mean of the medians to calculate each group. So we take the median percent tail DNA of each animal out of the 150 that are scored for each animal, and then we do means per group. So when at the end of the day, these folks are scoring 750 COMETs per dose group. There's 5 dose groups, usually 4 dose groups, whatever that math is. And then they got to go back and score another 100 cells for hedgehogs. Very labor intensive, one at a time scoring, archaic, and it ain't going to change for a while. We always do histopathology to show that we get a positive. If you have a positive response, we want to make sure that it's not due to cytotoxicity. So those are all the assays that are regulatory. How do you interpret a clear positive? Well, you got to have a statistical increase, a dose response, and finally, you have to exceed the laboratory historical negative control data. Those 3 are critical for in vitro tests. So when all criteria are met, this test chemical is positive. When none are met, it's negative. When 1 or 2 of these are met, it's equivocal. And I can tell you, clients hate that. We have had a number of situations where we get a nice positive control. We get a nice positive response statistically. There's a dose response, but it does not exceed our historical controls. It could be that day the background was a little lower. And so now we have a situation where we have dose response, we got a statistical increase, but it does not exceed our historical control. So right now we were saying those are equivocal and they're not biologically relevant. So that's what we're using now. When you go in vivo, however, those same criteria are needed, but when you have, so positive, no one cares if it's positive, but if it's negative in vivo, you got to go in there and show tissue exposure. One of the tests we've done recently for a client that was Mike Stryker ran it, we had it was negative in the 3 tissues we looked at. We had to do bioanalytical on the 3 tissues, liver, nose, and I think it was blood, for the parent compound and a metabolite to show that this test article was absorbed into the animal, was systemically available, and the tissues were exposed. It's got to be done bioanalytically, and it's kind of bad because when I tell a client, hey, your response is negative in the in vivo micronucleus, the bad news is you have to spend another $30,000 to come up with a analytical method to demonstrate that your compound was in the liver or was systemically available. Follow-up test for a positive, particularly for in vitro positive, go directly in vivo. That's what my recommendation always is. Go in vivo for a lot of reasons. Drug metabolism is the main thing. As I mentioned, S9 has phase 1 metabolism, not phase 2. It's not how cells work in vivo, not how anything works in vivo. Phase 1 and phase 2 metabolism are linked. It's a one sequential, next one to the next. So when you disrupt that, you're going to have a bunch of reactive metabolites that got nowhere to go except binding DNA. And this is one reason we got this SBIR grant, because we're using HepRG cells, which have both phase 1 and phase 2 metabolism, and that COMET chip technology I talked about with MIT to develop high throughput COMET assay in human hepatocytes. There's also vivo test to follow up. One is the big blue mouse assay, and I used to beat that to death back when I was a post at CIT. However, now BioReliance was slick and they bought all the rights to the big blue mice. So if you get a situation where you need a transgenic mouse mutation assay, you got to go to BioReliance. You don't have a choice. One of the problems with these assays, the big blue assay, completely bind to point mutate, to chromosome damage. So if you do the big blue mouse, you're only going to detect point mutation. So it's the equivalent of running an Ames test. You can't detect chromosomal damage, it just doesn't happen, and there's a lot of reasons why. You can talk about it if you like. More recently, the flow cytometry-based PIG-A gene mutation has been developed. Myself and Cheryl Hobbs are on the OECD expert working group. We just put together the final version, which was going to go through review within the group of the OECD group, and then off to the OECD WMT, which makes the decisions regarding assay, validation of the assay. So negative in vivo test, in vivo genome, we have so the in vitro. So you go negative in vivo, you got to be able to do tissue exposure. You got to demonstrate that the compound is available to the systemic circulation. And for the common assay, you got to do tissue levels, histopathology, some kind of method that you can use. Typically, it's a bioanalytical method to show that the compound is in the tissues, it reached the tissues, and was absorbed. So you have a positive in vivo genotoxicity test. You want to make sure that they meet all three requirements. You want to make sure that it exceeds historical controls. In the micronucleus assay, you look at percent reticulocytes because that's kind of a measure of toxicity for the micronucleus assay. In the common assay, you do histopathology because you might have a complication due to cytotoxicity in that tissue causing some side effect DNA damage. You can also determine benchmark dose nowadays and do TTC to show that humans will never be exposed to this concentration. If you're in drug world, that will kill the drug, basically. And by the way, when I worked at Merck, a positive Ames test would kill a drug. There were no drugs even at that time. Even for chemotherapeutic drugs that could usually treat cancer, you weren't allowed to have one that's Ames test positive, even if like we were testing coccidiostats and for chickens and for Merck, and they no Ames test positives. Just to wrap up, there are some newer assays coming up. Again, you can do all these on this particular screen here. It doesn't help you with respect to getting your drug on the market. A lot of these are used now for what's referred to as drug discovery, not preclinical safety assessment. Two big issues. The high throughput assays kind of thing, that's drug discovery, not preclinical safety assessment. Some of them are being transitioned. The genomic marker down there, the very bottom in blue and red, is something we've done here. We just, Carol Yock, it's been great working with her. We've been able to train like maybe close to about half a dozen postdocs on different assays using our collaboration and HepRG cells. And she and her colleagues validated this TGX DDI biomarker, which is a P53 DNA damage response biomarker that can be used, and it's passed through FDA to be a biomarker that can be used. But again, it's not in that GeneTox test battery I showed at the beginning. And, basically, you can do this, get an idea, but for preclinical safety assessment, drug world, it's not useful. It is useful for research, EPA, those kind of folks. Future directions, reduce reliance on animals. Again, this is how we got a grant here using HepRG cells. Toxicogenomics, there is a lot of effort on gene expression profiling to develop benchmark dose. And finally, the analytical detection of rare mutations. As I mentioned earlier, mutation assays take forever to do. You got to treat cells, you got to wait for expression to occur, blah, blah. It takes a long time. With this new technology developed by Twin Strand Biosciences called error-corrected or duplex sequencing, it can replace the big blue because it enables the determination of pulling out that 1 in a 100,000 and a 1 in 1 million. That's depicted on this slide. And this is kind of like my next to last slide. So one of the questions was, how did I end up in RTP? Well, when I was a graduate student back in 1986, I saw this presentation by a fellow named Tom Skopek on the left-hand side. And he was showing that if you sequence mutations in a sequence of DNA, this is the C2 gene of lambda, and you treat with different chemicals, each chemical, at least shown here, has its own characteristic mutational spectra. So in other words, in the stretch of DNA, you have a spontaneous pattern, UV pattern, and these are all different and distinct from what you see spontaneously. And this is all DNA sequencing, and it's done clone by clone, one at a time with mutations. And so my lab at CIT did this. This is what we did for a long time, working with a number of compounds. And nowadays, 2020, it's over here. You can do direct analytical assessment of mutation using next-gen sequencing. And so when you look at a stretch of DNA, like over here, this is a stretch of DNA, 400 base pairs long. If you take that and put it on a DNA sequencer, you get all these mutations because it's been amplified, you get all these errors everywhere, fragments, so all this is a big noise. However, this company called Twin Strand Biosciences basically have developed the bioinformatics tools to pull out the true mutation. So this is a technological leap. We tried doing this. I tried doing this, many years ago. We just didn't have the technology. And sequencing back then, you can tell 50-50, but you couldn't tell much beyond that. Nowadays, you can pick up the 1 in 100,000, 1 in a million using this technology. Finally, you can do a lot of this in humans. You can go directly into humans, measure all these events shown here. There are a number of confounders, and this has been used in some compounds like benzene, butadiene. But again, you get a number of confounders. And then just to end up, so you guys have an idea about cost, So when we run an Ames test, for instance, it's about a $9,000 assay. However, because I got asked this about a screening assay. Well, when you do the analytical portion of it, so when we do a GLP Ames test, it costs $9,000 for the Ames test. But then you also have to have a validated method to quantify your dosing solution. That's another $20K. So now you're going from a simple test that is $9,000 for GLP compliance. You got to measure the solutions analytically, and now it's a $30,000 test. So that's so these tests can be used. The Genotoxic test package is about $80,000, $90,000 test when you include analytical.